Good morning, good afternoon, and good evening, everyone, wherever you may be joining us from. My name is Ligia Prodan, and on behalf of MDPI and Nutrients, I would like to warmly welcome you to today's webinar. Thank you for taking the time to be here with us. At the beginning, I would like to share with you some information about MDPI. We support academic community since 1996 with more than 400 journals. MDPI is based in Basel, Switzerland, and has the mission to foster open scientific exchange in all forms across all disciplines. MDPI is committed to provide high-quality service for our authors. Nutrients is an international, peer-reviewed, open-access journal dedicated to advancing research in human nutrition. It focuses on publishing novel insights into the impact of nutrition on human health, as well as innovative methods for assessing nutritional status. The journal also welcomes manuscripts that describe the outcomes of animal studies, provided they are relevant to human health. We have three great presentations for today, and they are followed by a Q&A session. As a reminder, today's session will be recorded for those of you who wish to revisit the material later. We have a relevant special issue titled Nutritional Therapy of Intestinal Diseases and Intestinal Microecology, with a deadline for submissions of 25th of June this year. Please feel free to submit your questions at any time using the Q&A function, and you can find it here. Now, without further delay, I am pleased to introduce you the chair of this webinar, Professor Chi Su. His leadership and expertise will ensure a stimulating and productive session. Professor Chi Su, the floor is yours.
Dear professors, colleagues, and online participants from all around the world, good morning, good afternoon, and good evening. This is Chi from the Chinese University of Hong Kong. On behalf of the organizer, it's my privilege to welcome you to today's webinar, Nutritional Therapy of Intestinal Disease and Intestinal Microecology. We are honored to host speakers who have shaped the very foundations of microbiome research and nutritional therapy. Please join me in welcoming our first speaker, Professor Jin Wang, who will guide us through the fascinating interplay between food allergy and gut microbiota. The floor is yours, Professor Wang.
Okay, thanks. Thanks for the invitation from Professor Su and Professor the Nutrients webinar. So give me this chance to have a talk with all the friends from other countries and even from different universities. So right now in mainland of China, it's in the evening. So I'm saying hi from Nanjing to everyone. So I'm going to share my screen. Just wait a moment. So like the Professor Su mentioned, my presentation title is about food allergy and gut microbiota. So right now, from the data from the Food Allergy Organization and World Health Organization, they reported there around 6 to 10% of the global population are suffering from food allergy. I don't know if there anyone allergic to any food, even the dust and the flowers and other kind of stuff. So if you guys have this problem, you can tap it in the chat area, so we can talk about it later. And there's a big 8 and from all over the world, and they're the main food allergens include the milk, eggs, the peanuts, and seafood, and other food. And there's a new thing we have to pay attention. Since the last year, the sesame has become the new big 8 family members and replace the soybean. However, in China, there's a little bit different. And most of people, they're allergic with the wheat, seafood, egg, milk, and even the fruits. Thus, a food allergy has become one of the challenges for the global food safety and public health. We can see from the map clearly from different countries, they have the problems with the food allergy. And also there's a data since the 1999, and there's the food allergy rate, since 1999 and until 2019, and there's around 3 times higher, from 3.5% to 11% in the infant. So there's really a problem in the children. And in our group, we also did a little bit work about the food allergy in China. And we found in the north and south, they're totally different. In the north, and most of people, they're allergic with the nuts, milk, eggs, and fruits, and wheat. However, in the south of China, a lot of people, they're allergic with the seafood, because over there, they have a lot of seafood can be obtained and for the regular foods. So they're totally different. We can see from the map. So from the south and north, they're totally different. And for the simple terms of food allergy, there are 2 kinds of simple terms. And one is the mild simple terms, include the itches and the swelling of the lips. And however, there are also a lot of serious simple terms, like difficulty breathing and shock and even death. So we can see the different simple terms from the body. And there's an example and from the movie, and this is the mild simple terms of the food allergy. So this is the mango allergy. So after consuming the mango, so you can see the lid is itching. And also we have the serious simple terms from the food allergy. And this story happened in Canada, Montreal, and this lady was killed by the peanut allergy. So all over the world, there are a lot of challenges for the food allergy, because we don't have effective treatment for the food allergy. We can only to avoid the allergen from our daily food. And also we don't have enough service for the food allergy, especially in the mainland of China, because there's a lot of people care about pay attention to the food allergy. So this part is really need the data for the food allergy. And also for the food allergy assessment especially for the entire food chain is not perfect. And we need to increase the process of the detection of food allergen. And for our group, we did a little bit work about the probiotic anti-food allergy. And we worked with in a Mongolia university, and we selected several probiota from the healthy breast milk. Because for now, a lot of people, they talk about for the baby, when we give the baby breast milk or the formula milk. So this is the topic. And we successfully separate probiota M9 from the breast milk. And we did several studies. And the first study is we try to know the effects of breast milk and the M9 on the food allergy. And we built the food allergy model and used the OVA. And we used the M9 to treat the model, and we want to know if this probiotic can improve the food allergy. And our results found that M9 can maintain the body weight compared with the control group and the OVA group. And also the M9 treatment can reduce the diarrhea scores and reduce the histamine and MCP and also the IgE level in the mass and also the mast cells. Then we also determine analysis the inflammatory stuffs from the mouse, and our result found that the M9 probiotic can improve the IL-4 and IL-13, the inflammatory, and also can change the Th1 and Th2 balance and towards to a Th2 response. So for the M9 probiotic, we also found it can improve the food allergy from the microbiota level. And first, we can see the M9 can increase the CHO and ACE level and also increase the microbiota abundance, especially for the F and B ratio. And also the short chain fat acid producing is also increasing. For the SCFA, so we can see the butyric acid is becoming very high compared with other acids. So after the M9 probiotic treatment. So in the end, for this first study, and we found the milk, human milk probiotic really can improve the microbiome and immune status and improve the food allergy symptoms, such as the gut metabolic, especially for the butyric acids. However, we want to know if really the short chain fat acid can regulate the food allergy. And then we did the second studies, and we want to know if the effects of short chain fat acid, especially the butyric acid on the food allergy. So for this study, we collected some samples from the healthy children and also the food allergy children. And we got some, we did a little bit analysis from the samples. We found there's a big difference between the healthy children and the food allergy, especially in the CFA level, especially for the butyric acid level. So here we can clearly see, and the butyric acid is the most efficiency in the improving the food allergen among all the SCAFS. And in the animal experiment, we set different groups, and we use the SCFAS include the butyric acid. And from the tissue analysis, we can see the butyric intervention can improve the values head and also can restore the globulate cell numbers. And also from the tissue analysis, we also found the Batrate treatment can upregulate the ZO level and also the MERK2 expression. And from the PCA analysis about the microbiota, and we found there's a big difference, especially around the OVA group and the Batrate group. In the OVA group, we can see clearly, and in the mouse, there's a reduction in the FB ratio, and also abundance of the microbiota is also decreased. But however, in the Batrate group, and after the treatment, and we can see the FB ratio is increased, and also enriched the microbiota in this group compared with other treatment and the control group. And we also analysis the effect of Batrate acid on the gut metabolites, and we found there are some components, especially the anti-inflammatory and the antioxidant compound, and in the samples is increased, and also the tryptophan component is also increased, and this component can significantly inhibit the beta hexosaminidase release. So it really can help to improve the food allergy, simple terms. And also from the PCA analysis, we found after the treatment, especially the Batrate acid treatment, can downregulate the immune regulatory and the inflammatory, like the IL-4 and 13, as we mentioned before, and also can suppress the Notch signal pathway and include the expression level of the key genes like the Notch1 and the Jungle D1, and also the HES1 in the Notch pathway. And we found this kind of affection is related to the Batrate acid, and it's related to the ROS response. And we found the Batrate acid treatment can reduce the ROS accumulation and enhance their antioxidant capacity. And also, the data also showed, and the Batrate acid treatment can upregulate the tight junction proteins, like we mentioned before, like the ZO1 and the MOX2, the expression. And also there's a question if all kinds of those Batrate acid can increase or improve the food allergen, simple terms, and the answer is no. And we did the dose response effect of Batrate on the ROS stress and the Notch signal. And the result found that, and for example, the Batrate acid, when the level is 0.25 to 1 molecular suppressed, can suppress the ROS and restore the antioxidant level, while the 5 the Batrate acid cannot exert the antioxidant effects. And also for other kind of, for example, the MR expression, and also the ZO1 and the eco-herin the MRL level, we did the same experiment. We found there, a different response related to the contribution, the level of the Batrate acid. So for these two experiments, we found the probiotic M9 and from the health breast milk can really improve the symptoms in food allergy. We use the mouse model, and also from the clinical level, and from the fecal samples we collected from the children, and we found they are really different, especially there's a significant reduction in the food allergy children of the SCFA level. And also we use the animal model to analyze the budget treatment, if it can really increase the SCFA level in the animal models. And for the molecular mechanisms, and we found the butyrate acid can regulate the level of the microbiota, and also the ROS notch access in the cells. So all the data we published in the last months in the journal of Meta. And if you guys are interested in this paper, you can download it from this website. And there's a short introduction for our research team. And right now in my research team, we have 2 professors and 2 associate professor, and 1 lecturer and several postdoc, and some graduate students. And right now we also cooperate with the hospitals like the Nanjing Children's Hospital and other like hospital. And also we worked with the Jiangnan University and McGill University and the University of Auckland. So I would like to thank all the funding support, and also the other professors to work together for this job. And if anyone are interested to know more about our research, and can scan the WeChat to have a detailed talk. And except for the food allergy, and in our lab we also did some 3D printing research, and also the diagnose based on the AI. And in the end, I would like to welcome you guys to visit the Southeast University in Nanjing, and hope to see you in Nanjing. Thanks.
Thank you very much for your excellent presentation. It's very interesting to see the probiotic and also the short-chain fatty acids can help elevate the food allergy. Okay, it's time to move to our second speaker. Now let's give the stage to Professor Fu Yongli from Zhejiang University.
Thank you so much for your introduction, Professor Su. And it's my great pleasure to present my research. And today, I would like to share our recent research, cardiometabolic benefits of a non-industrialized type diet are linked to the gut microbiome modulation with you. Firstly, please allow me to briefly introduce myself. Right now, I'm a professor in animal breeding and genetics in Zhejiang University. I obtained my bachelor degree from mainland China. Then I conducted my master program in Europe and received double master degrees from both Wageningen University in the Netherlands and the University of Natural Resources and Life Sciences, Vienna. After that, I joined the University of Alberta in 2012 and achieved my PhD degree in animal science in 2017. After getting my PhD degree, I conducted research projects on human probiotics and nutrition for more than 4 years as a postdoc. Then I joined in CTU as an assistant professor in animal nutrition and worked in Hong Kong until September 2023. Now I would like to talk about this study. Industrialization depleted our gut microbiome and caused the decrease of microbial diversity, lower ability of fermentation, reduced production of short-chain fatty acids, less enzymes for plant-based carbohydrate degradation, and so on. This negative influence has been associated with increased incidence of chronic diseases and immune disorders, for example, diabetes. To face this challenge, a strategy that supplies the lost microbial taxa and providing their nutritional support at the same time might be the best hope for the microbiome restoration. This may restore the missing features of our gut microbiome and thereby improving our health. Why did we choose allergy? Because species are affected by the industry regularly detected in the ago, but is rarely found nowadays. In our previous study, we found that they are frequently present in the non-industrialized Papua New Guinea population. I will call it PNG for short in today's presentation. Well, they have been considered to be missing from the industrialized US population, as shown in this figure. Meanwhile, this species is generally recognized as safe and has well-established healthy promoting effects, and therefore we don't need to worry about the ethical and safety issues when we reintroduce it to the humans. Here, I would like to provide more specific information for this designed non-industrialized type diet. We designed this diet, call it Restore diet, based on the dietary pattern of rural Papua New Guineas. When we designed this diet, we also considered the availability of food ingredients in Canada. In other words, we selected food that are commonly consumed in Canada. And this designed diet has a high amount of polyphenols and stachyls that are the substrates for the allergy. And this diet was plant-based with minimum processing, which is rich in whole grains, fruits, vegetables, legumes, and nuts. And this diet has a high amount of fiber, but limited amount of animal protein. Well, we didn't include any dairy or wheat products because they are not consumed in PNG. And here is an example of the daily food of PNG population. This is the sweet potato of allergy and also the gut microbiome and host metabolic status in healthy Western adults. To achieve our objective, 30 participants consumed either their Euro diet, as shown by this orange color, or this designed non-industrialized diet, as shown by this green color, for 3 weeks. And after washout, they switched diet for the second period. A single dose, you can see here, as shown by this cup, a single dose of L-reuteri PBW1 strain or the L-reuteri type strain DSM-20016 or the placebo was provided to the participants at each diet period. And this L-reuteri PBW1 is a strain isolated from rural Papua New Guinea, which has quite genetic dissimilarity as the type strain DSM-20016, as shown here by these ANI values. And after that, multi-omics approach was applied to characterize the gut microbiome and to evaluate the host metabolic status. We performed the quantitative plating using L-reuteri selective media and strain-specific qPCR to estimate the persistence of L and the survival of L-reuteri. Both L-reuteri strains declined and become undetectable after 17 days, with exception of PBW1 in one subject. And you can see here from this graph, from this figure, the designed restore diet enhanced the short-term persistence and survival rate, you can see here, of PBW1, especially 4 days, you can see here 4 days, and 8 days after taking the L-reuteri. While no diet effect was detected for the type strain DSM-20016. Meanwhile, single dose of L-reuteri didn't show any effect on host phenotypes or gut microbiome. During conducting this study, we realized there was no standard protocol to design and validate strain-specific PCR-based approach for the absolute quantification of bacterial strains in fecal samples. Therefore, we systematically compared qPCR and droplet digital PCR for the absolute quantification of L-reuteri strains in human fecal samples and developed an optimized protocol for strain-specific primer by this graph. And this study has been published in Microbiome last year. By using the samples collected from our clinical trial, we quantified the L-reuteri PBW1 and DSM-20016 using qPCR, RNA gene sequencing and selective culture, as shown in this graph. For 16S, you can see here, and metagenomic the number of could reach 10 to the power of and to abundance material in the gut. These two methods are not that sensitive. Could you hear me, Professor Su?
Yeah,
I think
it's
my
okay now.
internet was
Yeah,
was
it's not
interrupted
very.
for
Yeah, it's not very stable, but should be okay now.
Okay, sorry for that. The network is not that well from my side.
You can.
Apologize for that. So I would like to continue shifted the gut microbial community structures according to the PCOA analysis. But microbiota continued to cluster by individual, you can see here, and each color represents one individual. And the percent of microbiota variation diet can explain the diet you can see here from this table. Within each individual, the diet can explain from 22% to 58% of microbiota variation, suggesting a strong individual effect of the restored diet on gut microbiota composition. Our restored diet also decreased the alpha diversity indices, such as the Shannon index and observed features for both compositional and functional features. And the restored diet induced higher beta diversity differences for both compositional, as seen here by the ASVs and SGBs, and functional features when compared the microbiome with baseline of each individual, meaning the diet significantly affected the microbiome composition and functions. When we focus on the relative abundance of each microbial taxa, we found that the restored diet altered more than 50% of detected ASVs, and several healthy promoting species and genera. For example, the genus Bifidobacterium with 3 Bifidobacterium species, for example, the Bifidolangum, were enriched during the restored diet period. And the relative abundance of pro-inflammatory bacterial taxa, such as this Brophia and Ruminococcus and Streptococcus, they were reduced. The restored diet increased the ability of gut microbiome in carbohydrate utilization through enriching the enzyme genes, especially the enzymes for plant carbohydrate utilization. And meanwhile, the fiber fermentation of gut microbiome was enhanced by the diet, supported by the increased total short-chain fatty acids, especially this acetate, as well as the reduced pH value. Also, the protein fermentation of gut microbiome was inhibited by the restored diet, as shown here by the decreased the amount of branched chain fatty acids. After that, we analyze the resilience, stability, and the interactions of microbiome during the diet intervention. We found that the restored diet shifted the microbiome super fast. After 4 days of the intervention, both alpha and beta diversity started responses, and then maintained this status. You can see here, they maintained this status during the intervention. But once we stopped the intervention, the gut microbiome returned to baseline status by the end of the washout. We also detected more positive correlations between gut microbes and higher stabilities during the diet intervention, as shown here. meaning our restored diet enhanced the collaboration between microorganisms. Then we characterized the plasma metabolic profiles during the diet intervention, and we found a significant global effect of the restored diet on the metabolome, and 31 metabolites was significantly altered by the restored diet, and there were 22 increased metabolites, including these three indole 3-propionic acid, biotin, and peptolic acid that are considered as beneficial, while there are 9 decreased metabolites, including some bile acids that are considered as harmful to the host. The restored diet also caused a significant shift in metabolic markers, according to the PCA analysis shown here in this figure. When we zoomed in for each marker, we further confirmed our findings. Consuming this diet improved several metabolic and immunological markers, including the body weight and BMI, and CRP. This is a marker for the inflammation. Glucose is a marker related to the glucose metabolism and insulin, as well as different types of cholesterol, which are markers for lipid metabolism. And furthermore, the diet also reduced fecal protectin, indicate a decreased gut inflammation. Then we linked the baseline microbiome and shifts of microbiome induced by the diet with the metabolic and immunological risk markers of chronic disease using random forest models, and thus to determine the potential mechanisms for how the non-industrialized diet benefits markers of chronic disease. For glucose, we applied RF classification models because there are responders and non-responders to the diet intervention. For the other markers, we applied RF regression models because the responses of these markers to the restored diet are continuous. According to our analysis, we found that we could predict the response of healthy markers using baseline, so the healthy benefits, healthy effects of our restored diet are determined by each individual's baseline microbiome and metabolism status. And we can also predict the response of healthy markers using shifts of microbiome and metabolome features, meaning the benefits of our restored diet are mediated by the microbiome and the metabolome. In summary, the restored diet, the diet that we designed, it reduced the microbiome diversity, but enhanced the evolutionary persistence. It redressed several microbiome features that negatively affected by the industrialization, and induced beneficial changes to the microbiota-derived plasma metabolites. It also has profound metabolic and immunological benefits for humans, and those beneficial effects could be predicted by microbiome features. Our paper has been published in Cell this January. After 30 days, it was the most read paper when I checked it on February 20th. And the editors of Nature Medicine, Nature Review, Microbiology, Cell Host, and Microbe all reported our findings in their journals. I would say that we didn't apply the fancy technologies in this study, but we answered a question that many people are caring about using straightforward experimental design and approaches. That's why our study attracted some attention from other scientists. So thanks a lot to Professor Jens Waters for his support to conduct this project, and Dr. Anissa, who is a postdoc at the U of A, made significant contributions to design the diet and conduct the human clinical trials and measure the host metabolic markers. And her works and her contribution are sincerely appreciated. And many thanks to all the other members involved to this project. And we are grateful for the financial support for the funding agencies. Thank you so much for your time and attention. If you have any questions or feedback, we could discuss this later. Thank you.
Thank you very much for your interesting talk, Professor Lee. If possible, I really want to try the restore diet to help me lose some weight. Okay,
Thank
please,
you.
yeah, please join me welcome our last speaker, Professor Ren Gan.
Hello, everyone, and good morning, good afternoon, and good evening. I'm Ren Gan from PolyU, and first, I'd like to thank Professor Su's invitation and MDPI Publishers Organization. And today, I would like to share with you about a topic about the tea and human health that we previously did some work in the last several years. I think this is a good example of the concept that food as medicine. So my presentation will mainly include the following 5 parts. So first, I would like to have a brief introduction about tea. So you can see this slide. Actually, currently, the tea can be classified into 6 categories, including the green, white, yellow, oolong, black, and dark teas, and mainly based on their fermentation and oxidation degree. From the green tea is long-fermented, while finally to the black tea is fully fermented, and the dark tea we call the post-fermented tea. Okay, so this slide shows some representative teas in China, and actually in the Western countries like the US and UK, people mainly like the black tea actually, but in Asia countries like China and Japan, the green tea is very popular, plus some other teas like the dark tea. And I would like to just clarify 2 teas that may be confused. One is the oolong tea, the Wuyi Da Hong Pao. Actually, this is a oolong tea, but many people may think it like dark black tea, actually. And also the Tie Guanyin, many people may think it as a green tea. Actually, both are oolong teas. So currently, for the tea production and export, the main countries are from Asia and Africa, like China, India, Kenya, and Sri Lanka. While for the import countries, many are Pakistan and some Western countries like UK, US, and Russia. And it's very interesting why Pakistan is the most is the biggest country that imported the most teas. I'm not quite sure anyone knows the reason. Maybe we can chat later. And next, I would like to talk about the main bioactive components in tea. And tea actually possess diverse bioactive components. You can see here, polyphenols, tea polyphenols are the most dominant bioactive components in teas, like the tea catechins, EGCG, and also tea is rich in tannin compounds, besides, tea are rich in polysaccharides, as well as proteins, especially such as the selenium-rich tea proteins. And tea also has some very special amino acids, like the L-theanine is very special in tea, and also such as the glutamic acid and GABA. And tea, especially in some fermented teas, are rich in, we call it pigments, tea pigments, like in the black tea and some oolong tea are rich in some such as the theophylline and also the cerubigin, while in the dark tea, like the pu'er, it is rich in the theobromine pigment. And the tea is also rich in alkaloids. Maybe many people think that coffee is the rich in the alkaloid, like the caffeine, while actually many teas are also rich in caffeine. And that maybe that's the reason that some people may, after drinking tea at night, they cannot sleep at night, probably due to the reason of the caffeine in tea. And tea also has some other components, such as some volatile component, like also the serponium. So next, I will briefly introduce part of these bioactive components. The first is the tea catechins are the main bioactive compounds in most teas. Especially we call the EGCG, epigallocatechin reganate, is the predominant catechin in most raw tea leaves and also the green tea. And also tea also contains some other minor phenolic acids and flavonoids. Besides tea catechins, tea actually, part of the tea are rich in some tannin compounds, especially the elagitannins. Elagitannins actually contribute to the astringent taste of the tea. Like many people just drink tea, you can feel the astringent taste, right? And mainly due to the elagitannin compounds. And the main elagitannin compound in teas are the strychnine, this compound. As you can see in the figure, just some yellow tea and part of the dark tea, especially the raw pu'er tea, are rich in the elagitannins, while some oolong tea and some reprocessed teas have the lowest elagitannin compounds. For the tea polyphenols, actually similar to other polyphenol compound or flavonoids, they have very low bioavailability. And it means most of the tea catechins cannot be absorbed in the small intestine, and most of them will directly enter our colon after ingestion, and then interact with the gut microbiota. So gut microbiota play an important role for the metabolism of tea polyphenols, but the location is in our colon. And currently there are studies about how gut microbiota can mediate the metabolism of tea catechins. And this figure shows the metabolism process is very complex, and it involves diverse biotransformation process, like the hydrolysis and reinfection, lactonization, dehydroxylation, and oxidation. And the recent study shows that some key gut microbiotas metabolites of the tea catechins exhibit better bioactivity than the catechin precursors in vivo, like the PVL and PVA, as here the red circle included. These compounds have been considered as the real bioactive metabolites of tea catechins that play or contribute to the health benefits of tea catechins as well as tea. And besides the tea catechins, And gut microbiota also play an important role in the metabolism of tea, the elagitanin compound. And for the elagitanin compound, after the gut microbiota metabolism, it can produce a group of metabolites we call the uracils, especially the uracil A. Okay, this compound has been demonstrated with many health benefits, especially with its potential to fight against aging. And also currently there are some nutraceutics or food supplements have been developed based on the uracil A to fight against aging because it is a very strong mitochondrial, can induce very strong mitochondrial biosynthesis. But currently, the gut microbiota mediated metabolism mechanism of the T catechins and elagitanins are still not very clear, including what gut bacteria strains, species that are involved in their metabolism and the related metabolism target genes or gene clusters, and for instance, the metabolic enzymes and the real bioactive metabolites. Many of these mechanisms are still not very clear, and also it is currently one of my main research focus. And besides this, theobromine is a very special bioactive component in dark tea, such as pu'er, and it accounts for about 7 to 13% of the mass of the dark tea. But the theobromine is not a pure compound. Actually, it is complex. It contains some different components, like some polyphenols or polysaccharide and some lipid amino acid like that. And besides, tea is also rich in some polysaccharide. We previously did some work about to understand the composition and function of 12T polysaccharide from China. And it is interesting to find that in the raw polysaccharide extracted from the tea, the presence of the phenolic compounds and also the protein in the tea polysaccharide contributed to their bioactivity, such as the antioxidant and the antidiabetic effects in vivo. So here, currently there is still this contradictory opinion about what really contributed to the functions, especially the bioactivities of plant polysaccharide. And our recent data suggest that some small molecules, such as phenolic compounds, conjugated with the polysaccharide may contribute to significantly contributed to their bioactivities. And also we found the tea polysaccharide cannot really be easily digested, but most of the tea polysaccharide can enter the colon and interact with the gut microbiota. And on one hand, to regulate the gut microbiota composition and balance, and on the other hand, the gut microbiota can metabolize the tea polysaccharide to produce some short-chain fatty acids. So overall, just the tea polysaccharide can be considered as the potential prebiotics or food functional ingredients. So next, I would like to introduce about the health functions of tea. Tea has many health functions like antioxidant, anti-obesity, and inflammation, or some liver or renal or neural or gastrointestinal protection, and also some other health functions. And based on, I will briefly introduce about our work, especially our work about the antioxidant effect of tea. We mimic the tea brewing process and produce some tea solutions. And we evaluated their in vitro antioxidant effect, and we found the green tea has the highest antioxidant effect in vitro, while the dark tea and white tea has the lowest. And here I didn't show that actually On the other hand, the in vivo antioxidant effect can be quite different. In vivo, the dark tea actually has very high antioxidant effect, while the green tea actually has low in vivo antioxidant effect, but the underlying mechanisms are still not very clear. And next, we also compared the antioxidant effect of tea with some other natural products, some common natural products, such as mushroom or vegetable fruits, some food byproducts of flower or wild fruit or medicinal plant. So overall, generally, the tea category has the highest antioxidant effects than other natural products in vitro. And for the tea's antioxidant effect, its main antioxidant compound can be tea catechins, especially the EGCG compound. You can see its chemical structure. It has 8 hydroxy group, which contributed to the high antioxidant effect of the tea catechin, such as the EGCG. But on the other hand, tea catechins also have or exhibit the pro-oxidant effect in some cases. This can be mainly due to their induction of reactive oxygen species, RS production, both in vitro and in vivo, because their hydroxy group is very active. So when it has a high dose or concentration, it can perform the auto-oxidation process, such as this slide shows. And then this process can produce some superoxide and also hydrogen peroxide to induce the oxidative stress in cells and tissues. So, as you mentioned, the tea catechins has both antioxidant and pro-oxidant effects. So, how to differentiate their effects? And this can be related to their dose or their concentration. In the low doses of tea catechins generate, like in the nanomole or lower micromole level, they mainly exhibit the antioxidant effect to scavenge free radicals. And also in their medium doses, like generate in the micromole level, they can generate moderate levels of RS. And in the both low and the medium dose of the tea catechins, they mainly exhibit the beneficial health effect, like prevention of cancer and the decreased body weight gain and the metabolic syndrome, and prevention of diabetes and cardiovascular diseases. While on the other hand, when the tea catechin at a high dose, it can induce high level of RS. And in this situation, they can induce oxidative stress to damage cell or tissue and induce toxicity and also kill cancer cells. Actually, this mechanism can be also the mechanism of many anti-cancer drugs that can produce excessive RS to kill cancer cells. And we also did some work about the dark tea cell browning to understand whether the dark tea cell browning TB can alleviate the non-alcoholic fatty liver disease with obesity. And we use the Yunnan pu'er tea extract. And we did 2 experiments in animal models, is the preventive and therapeutic experiment using the high-fat dairy induced mouse model. And we found the TB exhibit excellent preventive and also therapeutic effect on the NAFD with obesity. You can just see this figure after the treatment with the TB, the mouse, there shows much smaller in the appearance. And then we next, in order to further understand the underlying mechanisms, we did untargeted metabolics study using in the serum. And we found that the serotonin pathway has been, or the tryptophan pathway has been significant change after the TB treatment. And finally, we found marker in the serum, that is the serotonin, that has been, can see here, it has been serotonin, it has been significantly increased after the TB treatment compared to the control group. So we further investigated whether serotonin can regulate some downstream pathways related to the fat liver and obesity. And we found just the serotonin can control some signaling pathways related to such as fatty acid oxidation and lipolysis and oxidative stress in the liver, as well as in the adipose tissue. And then we also investigate whether TB treatment can change the gut microbiota. And we found that after its treatment, the gut microbiome abundance and composition has been significant change, especially with the significant increase with the abundance of some beneficial bacteria, such as the Akkermansia and bacteroids and parabacteroids. This species bacteria has been significantly increased after the TB treatment. And then we further want to investigate whether the gut microbiota play a critical role in TB's anti-NAFD and obesity effect. And we use antibiotic interference and further with the fecal microbiota transplant experiment to validate the gut microbiota is very critical for the TB to regulate the serotonin-related signaling pathways and the hepatic steatosis in the liver. So finally, as a summary, our study found that the dark tea derived the cell browning could alleviate the NAFD with obesity, and the mechanism can be related to the regulation of the serotonin related signaling pathway and the gut liver axis. And for other health functions of tea, I make summary of this table. Generally speaking, currently most evidence support the beneficial functions of the tea on the cardiovascular disease as well as the type 2 diabetes from clinical, epidemiological, and in vitro evidence. But for other health functions such as cancer, obesity, Parkinson, Alzheimer's disease, and depression, for these diseases, there are some positive study results, but currently more clinical evidence is needed to validate their health functions in humans. And on the other hand, I mentioned tea has many health benefits, but tea still has potential health risks. One risk is that the dietary supplement of the tea extract or tea catechins may have the liver toxicity, because and on the other hand, okay, drinking tea is generally quite safe, and the dietary supplement of the tea catechin, in some cases, it may damage the liver cells to induce the increase of some aminotransferase increase in the blood, suggesting the damage of the hepatocytes. And the reason for the dietary supplement can induce the toxicity can be mainly due to just the one-time intake of a high dose or concentration of the supplement can significantly increase their plasma or tissue concentration of the compound. That concentration can be higher or over the toxic threshold for our cells and tissues, though they may induce excessive production of the RS, okay, that can produce the toxicity. But for the drinking tea, okay, our habit is drinking tea from frequently, okay, and this process, drinking tea process, can be very difficult. Generally, it's impossible to reach the toxic threshold. So it is that's why the drinking tea is very safe. But the dietary supplement of tea extract or catechins in high dose for one time may have the health risk. Okay. On the other hand, as mentioned, the tea is also rich in caffeine. So for caffeine, it has many health benefits, but also on the other hand, excessive intake of caffeine can have some side effects that can induce the addiction, migration, or influence the sleep. Also has potential side effects on the pregnant women, fetus, and children. So the caffeine is another concern for the health risk of tea for some persons. Finally, I would like to briefly introduce the development of tea products. Currently, and for the tea product, there has a trend to produce some traditional tea beverage from the traditional tea beverage to the functional tea beverage like this. The functional tea beverage is added with the L-theanine beverage that it is claimed for the promotion, the sleep and mood for people. And on the other hand, currently there's the innovation of tea packaging or show form. Like the first picture shows we call the 百家姓 tea. It's a pu'er tea, okay, just with a very special packaging or show form. And the right picture shows the Xi'an Fu Cha. It's produced the Chinese chess tea. So it's very special, can attract the interest of consumers. And also currently tea has been developed into such solid or instant tea beverages. It's a trend. And also the milk tea and very popular in young adult, but milk tea is not very healthy. It is not really related to the tea itself, but the milk tea is rich in the sugar and trans fat and also some other food additives that are not healthy. And in recent days, tea has been developed into such as the cow milk tea, and also the tea coffee called the cha ka is very in recent days is popular. And on the other hand, tea can be extract can be used to produce some tea-based foods such as tea chocolate, tea sugar, tea mooncake, and some others. Okay, and also tea can be integrated with some other such as edible TCM to produce some functional food. This picture shows some functional food in Xi'an Fu Cha Tang. And finally, tea ingredient can be used to produce some nutraceutics, such as the HCG-rich anti-obesity products and the dark tea extract-based anti-obesity and blood lipid lowering products, and also such as tea pigment like the tea sulfavin-based blood lipid lowering product and the L-theanine-based antidepressant products. So finally, for take-home message, tea is rich in diverse bioactive components, and drinking tea is a very good habit with different health functions and is overall safe. And tea can be developed into some novel food functional ingredients and also functional food. So finally, and I would like to deliver a concept that tea is the typical example of food as medicine. And finally, I would like to thank all my previous research workplaces and the teams as well as my collaborators. And finally, thank you for your listening.
Thank you for your interesting talk, Professor Gam. And now we have finished all three presentation, and we comes to the next Q&A session. I will got several questions from our audience, and the first question goes to Professor Wang. Based on your presentation, we can see the prevalence of food allergy is varying across different countries. For example, we can see a high rate in USA, but a low rate in Canada. What's the reason do you think behind this phenomenon?
Okay, this is a very interesting question. So actually, yeah, I think one of the reason is about the food culture, because in Canada and USA, and food culture is quite different with China, and like in Canada or USA, the peanut allergy is quite common, because in their food culture they eat the peanut, especially the peanut butter every day and with the bread. But in China, we don't eat the peanut butter paste every day, but we eat some like a peanut oil. But the peanut oil is quite different with the peanuts, other kind of products, and we use the high temperature to process the peanut to obtain the oil. I think that's one of the reasons. And the other reasons, and we're different kind of people. And I think it's from the genetic level, and we're different. And also there's a cross activity with the food allergy. And in China, you are allergic to like milk, and also you will allergic with other pollens from the flowers or from the trees. And USA and Canada, we have different like trees and different pollens. So that is why some food in Canada is quite easy to allergic, but in China, even nobody allergic to this kind of food. And and maybe also there are other kind of reasons, and there need more researches to know this why. I think it's a quite an interesting topic for this question. Yeah.
Yeah, thank you for your answer. I think it do explain the question. And the second question is also about your probiotic experiment. The audience wants to know there are several commercial available probiotic all over the world, and some of them contain lactobacillus, but they are not the M9. Will those kind of probiotics can also help with food allergy?
Yeah, actually, the probiotic from the breast milk is quite rare. There's doesn't have a lot of products on the market, and right now we're working with another like university, in Mongolia University, and they have their own. The how to say, the probiotic, and because they have their special place to obtain this kind of probiotic. And actually this probiotic is already on the market in China, so you can buy it online. But from other countries, I also searched, but I didn't see any from the breast milk. I think this is a new probiotic, and especially for the patient from China, I think we can try for the children. And I guess maybe in different countries like in Canada and USA, they have their own like probiotics. And I will say you have to eat it every day, like because some probiotic cannot stay in the gut system forever. So you have to eat like every day and last a very long time. And to what I mean, maybe in the gut you can separate this kind of microbiota. So it takes time. So to get the the results to improve the food allergy.
Okay, thank you. I have a follow-up question. What's the special advantage of the breast milk derived probiotics when compared to the probiotics isolated from food or human feces?
So, sorry, you said the advantages.
Yeah, the special advantages of the amnestrin isolated
Okay.
from the breast milk.
Okay, from the milk, yeah, because from the milk you can get a very low like a concentration, but if you have the probiotic, you can have a very high concentration at once a time to consume, and maybe we will have a better result to improve the symptoms of the food allergy. But from the breast or from the food, it takes a long time and takes a lot of food to consume, and you have the similar results. I think that's the way, and will be easier and convenient when you consume the probiotic. Yeah.
Yeah, yes, thank you. And the following question goes to Professor Fu Yongli. And the first question is that from your study, it's quite amazing that a short duration of 3-week restore diet have significantly effect on human health and metabolic. What's your suggestion on the duration of this kind of special diet? If I want to use this diet in my daily life, how long I should keep using it?
Thank you for the question. That's a very good question and very fancy question for me. To be honest, I think 3 weeks is good enough to see the healthy improvement. But I'm not sure if we take this diet for a longer time, you will see the adaptation for this diet. It means probably you will see the effect could be continue or it may become less obvious. For this question, I'm not sure, and it's our next step to validate the effective
Yes. Oh,
for this.
okay. And based on your description, and the Restore diet is mainly designed based on Canada food.
But from the micro perspective,
Seems that the signal is not stable. The second question is, based on your explanation, the Restore diet is mainly designed based on Canada food. If people from different countries want to repeat this diet, what's your general suggestion to eat?
Thank you. So the diet was designed not only based on the Canadian food ingredients, it was designed to reflect, to represent the Papua New Guinea population's dietary pattern. But when we designed this diet, we selected the food available in Canada because that study was conducted in Canada. But for different countries, I would say for most food ingredients, they are available in the supermarket. So when we select and define the food composition, we consider the availability, means most of the food can be bought, can be reached from the different countries. Even in China, we have provide a recipe for this food for different meals. Like they are available in Facebook and also other LinkedIn and Twitter. I think they are available. So if you are interested, you can find those recipes from those websites to buy those food and give a try.
Yeah, that sounds great. Okay, thank you, Professor Li. And the
Thank you.
last several question is for Professor Gan. For the first one is that yeah, your presentation shows that tea has significantly beneficial effect on human health. But for people who cannot sleep or have poor sleep after drinking tea, how can they benefit from tea?
Very good question. Thank you. Actually, yes, tea, because tea contains caffeine, that's why some people, if you cannot suffer from or resistant from the caffeine, you cannot sleep after drinking tea. But on the other hand, tea also contains another component, especially the L-theanine. That compound actually can be helpful for the sleep. So for those people, it may be better to directly to intake the L-theanine based food supplement or nutraceutics, but not directed to drink tea.
Okay, that sounds good. And may I have your suggestion on which kind of tea has the lowest level of caffeine?
Lowest. Generally speaking, as our study found because for different teas, mainly based on the fermentation degree, but the fermentation itself cannot really change or degrade caffeine. So for many tea, actually, they contain a relatively high level of caffeine, and the fermentation process cannot really change it. So currently, there are a type of tea called the decaffeinated tea. That means some tea caffeine are removed. So for those for people, if you cannot suffer from caffeine, maybe you can choose the decaffeinated tea or have a try. Yeah. Thank you.
Okay, and we have the last question. Can you introduce the best way to drink tea, such as the timing or the water, such as hot water or cold water?
Anyway, just from the extraction angle, the higher temperature of the water can increase the extraction efficiency. But on the other hand, it also will just degrade the bioactive compounds such as T-catechins because due to high temperature. So generally speaking, the recommendation for green tea brewing is about 80 or 90 degrees, but not hot water or boiling water to brew the tea. And similar for other tea, I think.
Okay, thank you, Professor Gan. Although we still have a lot of questions from the audience, but it's time to end our today's webinar. And at the end, thank you all for joining us today. We hope you found the discussion as engaging and informative as we envisioned. As we move forward, let's carry the insight gained today into our practice, research, and discussion within our communities. The rising rate of intestinal disorder and metabolic conditions underscore the urgency of our work. Together, we can pave the way for innovative dietary strategies and improved health outcomes. Thank you once again to our speakers, participants, and everyone involved in making this event a success. Let's continue to collaborate and push the boundary of what's possible in nutritional therapy and gut health. Have a wonderful day. Bye-bye.
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 Right.